All right. 5.1a. DNA and RNA are required to make proteins. All right. I'm sorry. Hit the wrong button. Why proteins? Why are proteins so special? Uh, Proteins are important because they make up the structure of a cell. And it's also, they do almost all the work in the cell. Different proteins do different things, and they're very important because of what they do. Oops, I didn't mean to say that. Do, get it? Ha, sorry. Um, all right, all proteins are a bunch of amino acids. Proteins are larger molecules, but they're made up of 20 or so amino acids that are found in the body. Now, these 20 different amino acids come together in different um, ways and different amounts to make up the thousands of different proteins that are found throughout the human body. So, amino acids are what make up the proteins. Cool. All right. All right. We're going to go ahead and talk about. We're going to unwind our twisted ladder. Here's our unwound twisted ladder. All right. Our DNA strand. And across, there's these bases. Now, this DNA strand is made up of nucleotides. In the sidewall and these rungs, say this is our twisted ladder, our ladder, these sidewalls, this right here, this and these are nucleotides. And these nucleotides make up our DNA structure. Now we have four different types of nucleotides, okay? And they only go together in a certain way. Let me do this. All right, there's adenine. Now let me do it in the, yeah, adenine, thymine, thymine, cytosine. Oops, forgot to hit enter. And um, guanine. All right, these are the four nucleotide bases that make up our DNA rungs. Okay, and what's cool about these are I'm sorry, that these could only go. Let me see how to do this. How do I do this? I want a highlighter. There we go, highlighter. Okay, adenine and thymine can only go together, and cytosine and guanine can only go together. All right. So let me see here. So if this is adenine right on this base, then this one has to be a thymine. And if this is a cytosine, this one has to be a guanine. You can't have a guanine up here or a cytosine up here. A C goes with G, A goes with T. That's the only way that these can work together. That's the only way that they can fit. There's special structures in it. Um, these nucleotides that allow only an A and T to go together and only a C and G to go together. There may be like something like, maybe A, and I don't know this for sure, might be something like this, and T could be something like that. Um, Maybe that's how that goes, and then maybe the cytosine could be, heck, I don't even know. Oh, I don't know, a different form where this won't go in. Let me think. Maybe something like, um, maybe that's, and no, I have no idea. Don't ask me what that is. Maybe that's what the cytosine is. And the guanine would be a reciprocal of this, would actually have an opening that this would only fit in, not the thymine wouldn't fit in. So they're very different so that only A's can go with T's and T's can only go with A's. Okay, let me get rid of this. This is not going to be that easy, is it? Of course not. Okay, I got rid of it. All right, so all DNA goes through a process called replication. And all replication is where a DNA strand splits and let me see. All right. I'm going to twist this. I might twist this. Hmm. All right, I got it done. So basically, all replication is a process where the DNA strand splits. See these? C is G, G was C, T and A, C and G. C and G, and then here's the splitting. C over here's the G that went with it. T over here and the A that went with it. A. Do you see how they lined up? Okay, what's going on is these are just splitting right down the middle. Okay, and during this replication process, all of the DNA, basically this strand comes unwound and then splits. You have what's called a template. And what a template is a guide. If we draw a picture of a rabbit and we're able to, okay, back when you were in grade school, I'm sorry, little kid school, you put your hand on the, for Thanksgiving, you put your hand on the paper and you trace it to make the turkey. That is using your hand as a template. You're able to replicate something. So what's going to happen is you're going to put another C right here to go with that and a G right here to go with this. Okay, and right here would be another C. And right here would be an A. Well, you're not going to put it, your body is. And another A. And it's basically making another DNA strand. That's how you get a copy, a complete, accurate copy of your DNA. This is how it replicates, is once it splits, these nucleotides come around and fill in where the other one was. Now, it's not as easy as it seems. There's a lot of processes that we use or your body uses to get this done. And that uses RNA to do it. There's three types of RNA: messenger RNA or mRNA, ribosomal RNA or rRNA, and then transfer RNA, which is tRNA. Okay, so RNA, which is ribonucleic acid, carries information from the DNA to the ribosomes. Those ribosomes where amino acids are brought to make the thousands of different proteins. Okay, so RNA, and this RNA, let's get another page. We have three different types of RNA: mRNA, rRNA, and tRNA. Okay. Now each of these do a different thing. Transferring the information from DNA, from our deoxyribonucleic acid, to the RNA is called transcription. And transcription is just, it transcribes. Um, and those four nucleotides, okay, these, let me go to this. These three, or these four, adenine, thymine, cytosine, guanine, these four nucleotides are in DNA. Now, RNA has the same thing. However, almost the same thing. RNA has, let me see, three of the bases are the same. They have guanine, cytosine, and adenine. Gosh, forget to hit the enter button. Goes crazy on you. Now, there's still a fourth base, just like there is with DNA. But unlike DNA that has thymine, this has uracil. I spell it U-R-A-C-I-L. Uracil replaced thymine. So adenine and thymine go together in DNA. Adenine and uracil go together in RNA. Okay? So again, the four bases for DNA are different than the four bases for RNA. Three of them are the same: guanine, cytosine, which go together, adenine. In DNA, it's thymine. In RNA, it's uracil. And adenine and uracil go together. These RNA can make many copies of themselves off of one single DNA. So many copies of RNA can be made from the same gene in a very short period of time. Now, replication and transcription involve passing along information that's coded in nucleotide bases. To make proteins, cells have to translate that nucleotide bases into amino acid language. And how do they do that? I like the way the book does this. Well, first of all, how they do that, they use mRNA, rRNA, and tRNA. But the way the book puts it, you think it's DNA is a cookbook for making different proteins. It gives you all the directions you need. mRNA is a recipe for the proteins. It tells you what you need to make those. DNA tells you how to do everything. It's a cookbook. mRNA tells you how to make the protein that you're trying to make. Then the ribosomes is a place where the cooking gets done, the rRNA. It's where it all happens. tRNA then gathers all the ingredients, which in this case is the amino acids. So, and transfer RNA, or tRNA right here, does the actual translation of the bases to the amino acid language. It's like talking English and Japanese. You have an interpreter to translate it from English to Japanese. In this case, that would be the tRNA that does that, or the transfer RNA.